So this is what I kind of talked about a little bit before. DNA is deformable, and thus it can be packaged into chromatin in the cells to sort of be stored. It's going to wrap around histone proteins. So what is up next is an absolute crash course in transcription translation. What you will realize is that I'm going to skip almost all of the detail that you probably had to memorize in biology. I just don't care about that level of detail, all right? It is very useful if this is something you're studying, right? If you're in a research lab that's looking at different transcription mechanisms or translation, if you're really interested in this stuff, if you want to do metabolic protein engineering, absolutely, this is relevant. You should go take biochem. You should get into this stuff. For our class, because we do need to get into more of the thermo side of it, this is the speed version, okay? So the central dogma, which I think they're changing that language, they don't call it that anymore, of molecular biology describes the flow of genetic information in living cells. So replication, making copies of existing DNA in prep for cell division. That's our one-sentence definition of replication. Transcription, we're going from DNA to RNA. Copies of specific genes into a complementary strand of messenger RNA, or mRNA, okay, is our transcription piece. Next, we have translation of RNA into a protein. Okay. mRNA directs the assembly of amino acids into proteins in the ribosome. Okay. And the ribosome is a huge complex of other proteins and RNAs that I don't need you to memorize. Okay. So kind of the reason that this the central dogma, or this flow chart as it's usually drawn, is kind of going away is because we know that we now know that things go backwards, okay? And RNA is sometimes being reverse transcribed into complementary DNA in the cell. Again, not super important for this course, but if that's something that interests you, this is sort of the typical organization of how it's going to be shown, but we know that some other stuff can happen. It's a bit more complicated than this. But I mean, if you want to memorize the words in any way, the only difference I would really memorize is transcription is DNA to RNA, translation is RNA to protein. Okay. Here's where everything happens in our simplified looking cell. We have the nucleus. This is where we're storing our DNA. When we need to replicate DNA, we are going to have to uncoil it, open up the two strands, and we're going to form our mRNA here. So a good question that came up last time, as I said, polymerization of our DNA strands and our RNA strands is happening in that 5' to 3' direction. And then I said when we go to read these things, it was 3' to 5'. Then we had a great question about, I think RNA is actually read 5' to 3', and that is sometimes true. Correct, I looked that up. They're both always synthesized 5' to 3'. DNA will be read 3' to 5'. RNA, depending on what you're using, is it a tRNA? Is it an mRNA? Is it a functional RNA? Who's reading it and why? Where the protein binds and what it does? It could go either direction. I don't need you to memorize that. That's one of those things where it would be a true-false question like, is DNA read 3' to 5'? That's not important to me. So, but depending on what other courses you're in, it might be really important. Okay. So we're going to form our strand of mRNA in our nucleus, and then it's going to be transported out of the nucleus into the cytoplasm of the cell, where it will be the ribosome, which is this complex of RNAs, complex of many proteins coming together, simplified by salmon-colored blob, will bind to our mRNA strand. We have tRNA, or transfer RNA, coming in, and it is actually going to have this 3' anticodon sequence to code for a protein. And Monday is the protein structure conversation. Okay. But then we have this little growing strand of proteins, one amino acid at a time. DNA storage and replication. DNA is going to wrap itself around histone proteins, okay, into this this kind of hierarchy structure. We have just our DNA strand. It wraps itself around histones, and then they are able to pack themselves into nucleosomes, and then they coil up on each other. There is some chapters at the end of chapter 1 about supercoiling of DNA, actually it might be at the end of chapter 2. Either way, supercoiling will not be discussed, gene splicing will not be discussed. Okay? So if you see those sections, I won't ask you about them. Okay? So then we can have hypercondensed chromosomes storing our DNA. And then here we're showing DNA polymerase coming in and binding when we're actually wanting to make a copy. I'm not going to ask you about primers. I'm not going to ask you about leading and lagging strand. None of it. Okay? I just need you to know that once you look at DNA, who can bind to it and where and why, and that's where size and molecular recognition come in. Here are some definitions of these terms for you. If you'd like to have them, again, I'm not going to read them aloud to you. I recommend reviewing the slides or reviewing those sections that are relevant from the text if you're just not, you're not super fresh on some of this terminology. Other replication, I will ask you, again, I said I'm not going to ask you leading strand, lagging strand. I'm not going to ask you these Okazaki fragments, none of it. Okay. Transcription, we'll stop here and we'll look at it in a little more detail. It's similar to DNA replication because it is ultimately boils down to base pairing for that growing strand. However, there's a little bit of difference between replicating DNA and then making RNA from DNA. And the entire process is going to be regulated by transcription factors that have to bind to the DNA, and then they're going to recruit an RNA polymerase in. Again, this is a far oversimplified view of what this complex of interacting proteins actually would look like. Okay, but remember, we're reading from the 3' to the 5' end, and we're synthesizing from the 5' to the 3' end, okay, as we're making this transcript of mRNA. And once we've transcribed the gene that we care about, okay, again, I won't talk about promoting, how do we determine which genes are going to get turned on or when they're going to get turned off. We won't talk about silencing, any of that. That's for a bio class, a biochem class. Okay? Once you have the transcript of mRNA of interest, for our gene of interest, it's going to get transported out of the cell, and then the ribosome will take over. It'll match up corresponding transfer RNAs and produce the polypeptide. This is a really highly conserved process in all life forms, so regardless of whether we're talking about animals or humans, this process looks the same for everybody. What looks kind of different is some of the proteins that are involved in that ribosome, but ultimately the process looks the same. Okay. So unlike replication and transcription, where we rely on base pairing, we can't use that same one-to-one mechanism for translating mRNA into proteins. Okay, so rather it relies on this 3-sequence of bases codon. Okay? Transfer RNAs or tRNAs, they're very small RNAs that generally have this like clover-looking structure. Okay? They have an anticodon that recognizes a codon. So codon is a 3-base sequence. On the mRNA of interest, there's an anticodon, so it has our opposite pairs. On our tRNA, it will covalently link the amino acid in. So the tRNA comes in, binds to the mRNA. mRNA strand with the codon of interest, tRNA, amino acid. I think this slide just puts them all together. Okay. So here's a whole table of codons based on which base for RNA, A, G, C, or U, is in each position, the first position, the second position, or the third position. We have stop codons. What is a stop codon? What would be the opposite of a stop codon? A start codon, and we have some codons that actually code for stuff we care about. So maybe we have leucine, maybe we have isoleucine. Another thing I don't need you to memorize, I have no idea if they ever asked you to memorize this back in the day. I don't need you to know which 3 codes, like group of bases, matches to each amino acid. You would receive a table like this and be asked to use it, okay? So definitely don't worry about memorizing which ones are the stop codons. But we have codons that will code for things we actually care about, so our cysteine, our arginine, our serine, whatever it might be. Okay? So again, basic flow of how things happen. Biochemistry is ultimately built on the concepts of replication, transcription, and translation, and how the genetic code relates to the sequence of bases. and ultimately the resulting protein. Because the protein is generally what we end up caring about moving forward here. And that's why I don't care so much about this process of getting from DNA to RNA to protein, because moving forward in this course, proteins and how they fold and unfold and the pathological conditions they're tied to ultimately becomes more of our conversation, okay? So out of 64 possible codons, 61 code for our 20 amino acids. So there's some degeneracy and redundancy built in. Okay? What is a potential reason for having multiple codons for a single amino acid? Yes?
That way, if there's a mistake, for lack of a better word, tankable protein.
It's a great answer, perfect answer. This way, when there are mismatched or wrong bases in there that we don't want, some substitution has happened, oftentimes it's going to be okay and we're still going to be able to code for a functional protein. I already alluded to the fact that sometimes that doesn't happen. Sometimes a single base swap leads to a lot of bad downstream effects. But more often than not, building in this redundancy or this degeneracy makes it so that this process can be robust against errors.